Hey everyone. So we've been talking a little bit about DNA, but how do you replicate DNA, right? So we know a few things, like you are an organism, and organisms have offspring, and organisms heal and repair and grow. And so that means we need more cells for that. And that also means we need more copies of DNA in order to fill those cells and give us all the directions we need to make and maintain these organisms. So let's look at the idea of how do you make a copy of DNA. So DNA replication has to be really, really high fidelity, and it is for the most part. And it follows what we now know as the semi-conservative model. It's important because for a long time we weren't really sure. Did it conserve part of the old strand, when you were making a new DNA molecule? Would you have, maybe a parent strand, and then you just somehow made like a mirror image perfect copy, like non-conservative, or stuff like that. So we know now it's in this category here. So basically what happens are the two double helix strands separate. One of them becomes a template, right, or the example for the assembly or the copying of another strand, and that strand's going to be complementary, right? It's going to match up from all of these free nucleotides that are going to be floating around in the nucleus. We can use that. And then each new DNA helix has, again, the old strand right here, the parental strand, as well as that new strand, okay? Right. So here's what it looks like in a little more of an animated version. So we're going to unzip this parental DNA molecule. After we've done that, we use some mechanisms to insert brand new nucleotides that match. See, this is a C. Here, hold on, let's change color. Here's a CG pair on top. It means we're going to still have a CG pair. It just means here's going to be the older parental template strands. And this new one, this gray one, these are going to be the new, or the
daughter strands, so these little free nucleotides are going to be attached. Eventually what happens is the daughter strands are going to be generated from the parent strand, right? So this is a parent and daughters. Right? Okay. Even more closely we look at this is like the unzipping, right? So a molecule that it's actually this awesome enzyme that sits here, and it's called helicase. Okay, it's an enzyme that unzips DNA, okay? And it allows us to go from a double-stranded molecule to two single-stranded molecules, right? At that point, when we have the single-stranded molecules, we can start adding in those new nucleotides. But we don't just start in one place. Usually, at least in eukaryotic chromosomes, like yours and mine, we start in multiple places. When we're dealing with prokaryotes, right, so here's like prokaryotic, very double-stranded, we usually have like one area, for the most part, for gene replication. And that'll serve for that entire plasmid, or chromosome, the bacterial chromosome, prokaryotic chromosome. They have one, it's a circle. And it's usually not that a lot of information on it. However, we're eukaryotes, so we've got tons and tons of paired chromosomes, and we've got so much material we don't have to get all these linear chromosomes copied, right? So what happens is we have multiple origins of replication throughout our genome. What happens is they separate the daughter strand, the parent strands. Helicase pushes open this little bubble right here, the replication bubble, or the replication fork. Right. And the new strand is added on top of our template, our parental strand. And they push all the way until they find the roots, and then have the two full elongated molecules. Is directional as well. It means on one end, we have a phosphate sticking out. On the other end, we have a hydroxyl group sticking out. Yep, same story on this side. Phosphate over here, hydroxyl over here. We've seen OH. That's the 3' end. And then you see a P, that's the 5' end. That's important too. So notice they're anti-parallel, okay? They're going the opposite direction. So you add new nucleotides on the old or parental strand in a directional manner. We call this the directionality of DNA synthesis. So what happens is we have another molecule here, it's actually an enzyme. It's called DNA polymerase, right? And it grabs onto the 3' end and sits its butt right down, and it starts adding nucleotides in that 5' direction as it slides towards the 3' end. So, since we all came originally from prokaryotes, this isn't a problem if you have a circular genome. But now that we're eukaryotic and have all of these single-stranded chromosomes we've got to copy, we run into a problem when we start trying to replicate this other strand. It takes longer. It's called a lagging strand. The leading strand just follows this synthesis of 5 to 3' direction. However, the lagging strand has to actually jump on, sit on a primer, and then extend just a little bit, and then it falls off. And then it sits again, like it's another primer for 3', and then it extends a little bit until it falls off. Now, these are these little nicks and openings, okay? So these little nicks and openings just kind of you need to ligate them or stick them back together. It's all the way down this lagging strand, which actually takes a little longer. That's what we call this molecule here, DNA ligase. DNA ligase takes those bits and gives them a fix. There we go, all the way down. So the leading strand is the one that goes like the most canonical way. It's quicker, it's faster, there's no mix that you have to use DNA ligase to repair. However, the other side of the replication fork is the lagging strand, right, lagging. So it's the lagging strand right there. It's called the lagging strand because we have this extra step. We have to reprime and reseat polymerase multiple times, and then use DNA ligase to fill in any of those little gaps. Okay, so this is a really fun process, and you can actually take entire semester courses on how this operates. Because trust me, even though you're just going through a few tiny little enzymes, there's a whole lot more, and a whole lot more complexity. It's cool stuff, but you don't have to know it for our tests. Alright, I hope that helps.